So this is the third in the Chapter 5 set of videos. This video we're going to discuss cell signaling mechanisms. So there are multiple types of cell signaling mechanisms, and the simplest and most common form is local signaling, where a cell sends a signal to a cell that is next door or very nearby. This type of signaling is often referred to as paracrine signaling, and it occurs between many different types of cells. Synaptic signaling involves a neuron and its target cell, and usually that target is another neuron or a muscle cell. And this type of signaling involves a very specific type of junction between these two cells. The final type of signaling is long-distance signaling, in which a cell releases a signaling molecule that travels either through the blood or through other bodily fluids over very long distances. And this type of signaling is also called endocrine or hormonal signaling. But in each of these cases, one cell secretes a molecule that travels to bind to another cell and causes a reaction in the target. So there are multiple stages in most signaling processes. And in most cases, it starts with reception. It's necessary for the target cell to know that the signal is happening. And in order for that to happen, the target cell usually needs to have a specific receptor molecule for that signaling molecule. And in most cases, this is on the surface of the target cell. The next step in the process is signal transduction, or transmission of the signal from the membrane of the target cell into other parts of the cell. And this process often involves multiple molecules and multiple processes. The final step in the process is the response. And this can be changes in the cytoplasm, in specific organelles, or in some cases, changes in the nucleus involving genes being turned on or off in the receiving cell. So some systems are actually simpler than this, so let's look at some of those simple systems first. So one very simple signaling system is what's known as a gated channel. So remember we talked about channel proteins earlier in the chapter, and these proteins allow molecules to move passively through the membrane. But in this case, the channel isn't open all the time. It's only open under certain circumstances. So the gated channel works by binding to the signaling molecule. And a signaling molecule you'll find is also often referred to as a ligand. So the small molecule that binds to its bigger receptor target, the small molecule is the ligand, the target is the receptor. So the signaling molecule or ligand binds to the target, in this case the channel itself. That channel is embedded in the plasma membrane. The binding of the ligand causes a shape change in the channel, and that opens up the channel. In this case, we've allowed ions to flow through. And the ions themselves directly cause a cellular response. There are no intermediate molecules in this process. And this type of signaling is very important in neuronal signaling. Eventually, you need the signal to stop, and that can happen when the ligand either falls off or is actively broken down by enzymes that are in between the cells. When the ligand comes off, the gate closes again and the signal stops. So another simple signaling process is the one that is initiated by the hormone testosterone. So testosterone is chemically very similar to cholesterol. In fact, it's made from cholesterol. And testosterone is an exception to the rule that the hormone does not go inside the cell. So in this case, because it's so closely related to cholesterol, and we know cholesterol is an important component of membranes, this hormone can actually pass directly through the membrane. So it goes through the membrane, it goes into the cell. The testosterone receptor is actually in the cytoplasm of the cell. So the hormone binds to its receptor. That complex itself actually moves into the nucleus, binds to DNA, and causes changes in the DNA activating particular genes. So again, very simple system, not a lot of intermediate molecules, and unusually the receptor is in the cytoplasm instead of on the surface of the cell. So let's look at some systems that are a little bit more complex and maybe more typical. So this is a generalized signaling process, starting again with our ligand or signaling molecule, binding to its receptor, activating an initial relay molecule. And following that, there's a sort of series of molecules that get activated as a result. And you'll notice that each of these molecules that gets activated is a kinase. And kinase is an enzyme. We know that because it has ASE on the end. And in particular, kinases are enzymes that add phosphates to other enzymes or to their target molecules. So what happens here is that each of these kinases in turn activates another kinase. Eventually you activate some target enzyme that causes a cellular response. And this process is what's known as a phosphorylation cascade. So we're going to look at an example of signaling transduction, and we're going to look particularly at a system called the G protein coupled receptor system. This system is used in many different types of cells, in many different types of organisms, and many situations. And that's important because it tells us that this process, these types of molecules, evolved very early in evolution, and they've been sort of co-opted to serve lots of different purposes. So the basics of this system begin again with a signaling molecule. That signaling molecule attaches to its receptor. And in this particular case, that receptor is coupled to a G protein. Well, what's a G protein? It's a small protein that uses GTP instead of ATP as its energy source. And what it does is when the signaling molecule binds to the receptor, that activates the G protein. G proteins are tethered to the membrane. They are peripheral membrane proteins, and they're able to move along the membrane to get to their target enzyme and activate it and eventually cause a cellular response. So let's look at this in a little bit more detail. We're going to use epinephrine signaling as an example of a G protein coupled receptor system. And epinephrine, as you know, is important in responses to fear. So epinephrine is released, it binds to the G protein coupled epinephrine receptor. That activates the G protein. G proteins, in many cases, not always, but in many cases, bind to a target called adenylyl cyclase. And adenylyl cyclase is an important enzyme, also bound in the membrane, and its job is to make this molecule called cyclic AMP. Cyclic AMP is what's known as a second messenger in that it's a small molecule, it's not an enzyme, but it moves through the cytoplasm and can have lots of different types of effects. One of cyclic AMP's important targets is a molecule called protein kinase A, and this is what's going to initiate the phosphorylation cascade. Again, we have the theme of molecules binding to their targets, causing shape changes, and getting enzymatic effects as a result. So let's look at this in a little bit more detail as a complete system. So imagine the lion comes out from the bushes, you're afraid, lots of epinephrine is released. That binds to its epinephrine receptor. G protein coupled receptor activates the G protein. G protein moves through the membrane to bind to adenylyl cyclase. Adenylyl cyclase makes cyclic AMP. That activates protein kinase A. Protein kinase A then activates multiple other targets, actually, in fact, inactivating some targets and activating others. You get a phosphorylation cascade, multiple molecules activated. And at the end of this phosphorylation cascade, what you get is the production of glucose, which can then go into the blood and allow you to have enough energy to run away from the lion. So this system is one of the first major signaling pathways to have been worked out. The second messenger, the cyclic AMP role, was actually discovered by a guy named Earl Sutherland, and for that work he won the 1971 Nobel Prize. But as is the case in many situations with science, a lot of the pathway was still not very well understood, and in fact, almost 25 years after that, the Nobel Prize was won by Rodbell and Gilman for working out the rest of that pathway. One other thing I want to point out about this phosphorylation cascade, it seems like sort of a waste to have all these molecules in the middle. Why can't you just have cyclic AMP activate this enzyme and make a bunch of glucose? Well, the advantage of this cascade is that at each step, each of these enzymes can activate multiple copies of their target enzyme. So what you get is an amplification process, where for one or just a few molecules of epinephrine, you get thousands of molecules of active enzyme making lots and lots of glucose, so you can run really fast to get away from the lion. Finally, I would just want to briefly talk about the growth factor receptor example. This is a good example of a signaling cascade that results in changes just in this individual cell in the nucleus. So growth factor binds to its receptor. You get a phosphorylation cascade as part of the transduction process. As a result of that phosphorylation cascade, you actually activate a transcription factor. And what that does is to cause particular genes to be turned on in the cell. And this is a very common type of response. You can imagine if we've got a growth factor, we need genes that are going to cause that cell to grow and divide. And that is an introduction to some common signaling processes in cells.